Chapter 9 deals with the fundamentals of genetics, so we're going to be introducing genetics in this chapter. Chapter 9, Section 1 tells us what genetics is, which is just a field of biology that is focused on how characteristics get passed from parent to offspring, which is called heredity. One of the main people that we'll focus on is called Johann Gregor Mendel. He was an Austrian monk. He's considered to be the father of genetics, and he studied heredity. He was trained in statistics, was a monk, as I mentioned, and also a science teacher in the mid-1800s. And mainly he focused his experiments on his garden peas, which he worked on at the monastery. And peas provided him with enough opportunity and variety to see many generations over a relatively short period of time. So the characteristics that Mendel studied were 7 different heritable features, or things that could be passed down from one generation to the next. For instance, this diagram over here shows that peas can be either round or wrinkled. They can be yellow or green, have different pod shapes, different pod colors. The flower themselves can also be a different color, and then their stems can be arranged differently. So each one of those 7 characteristics had 2 possible traits, the yellow or the green color, the purple or the white color. So every characteristic had 2 different traits that he could study. His experiment is mainly 3 steps. The first one is where he started with 2 pure plant groups for a particular characteristic. Those pure groups are called strains, and this is going to be his pea generation, or parental generation. From there, he cross-pollinated them, which is a form of sexual reproduction, so combined 2 different plants together to produce the next generation. And this generation is called the F1, or first filial generation. And then finally, in his last step, he allowed the F1 generation plants to self-pollinate, which is a form of asexual reproduction, which doesn't involve a partner, and allowed them to produce the next generation, which is the F2, or second filial generation. And he did these crosses hundreds and hundreds of times and counted and recorded the traits of the offspring after every single step. And what he discovered was that whenever he crossed 2 pure plants in his pea generation, the F1 generation, or the combination of those parents, all had 1 trait. You never saw the second trait that was being crossed. But when he allowed those plants to self-pollinate and produce another generation, he started to see that trait appear again, and it was always in a 3-to-1 ratio. So what he discovered was that there must be a dominant factor and a recessive factor. The dominant factor is going to mask the other trait in the F1 generation, and then in the F2 generation, that 3-to-1 ratio comes upon us, and you start to see that other trait shown again. Fewer in numbers, but we see it appear again, and he thought that one must be caused by a recessive factor, one that's normally dominated by the dominant factor, or hidden by the dominant factor. So for instance, in this diagram here, he's got pure yellow peas combined with pure green peas. In the F1 generation, we only see yellow, so the yellow color must be dominant to the green color. And then in the F2 generation, we see this 3-to-1 ratio of yellow to green. And so that green color must be the recessive factor, the one that gets masked here in this F1 generation, but is able to reappear in F2. With our flower color, remember we had our F1 generation that was all purple, so the purple color flowers must be dominant to the white flowers. Mendel then came up with 2 laws based on all of his data that he collected, the law of segregation and the law of independent assortment. Now, the law of segregation occurs when a pair of factors gets separated during gamete formation, or sex cell formation, which we talked about in meiosis. So when those sister chromatids get pulled apart, one has one factor, one has the other factor. Could be 2 dominants, could be 2 recessives, or could be a combination of dominant and recessives, but they get pulled apart and separated and moved to the new cells that will be formed So that is called the law of segregation, that they get separated out when you form the gametes. And then his second law was the law of independent assortment. And we've also mentioned this one before. This is independent assortment is a way, along with crossing over, to get genetic variety in offspring. And basically, he said these factors that he was studying, these dominant and recessive factors for different characteristics, are distributed to gametes independently, which basically just means that the example I put up here says not all blue-eyed people have blonde hair. So just because you have one recessive trait doesn't mean you have all recessive traits, or because you have one dominant trait means you have all dominant characteristics. You get variety and different combinations depending on which egg cell of your mom's ends up getting fertilized by which sperm cell of your dad's. So it's a random and independent process. So this also is something that happens during meiosis. And molecular genetics is another branch of genetics that focuses more on the actual structure and function of chromosomes and each individual gene. So instead of just looking at overall characteristics and traits that are passed down, they actually want to know what each part of the chromosome does, what each gene controls. And then this is also where we have converted the name of Mendel's factors to alleles. So a gene is a segment of DNA that controls a particular characteristic, and all alleles are the two different forms of a gene that you could have. So these terms alleles are just replacing Mendel's factors. So they're still dominant and recessive, and we're going to represent dominant alleles with a capital letter and recessive alleles with a lowercase letter.